Okay, my slide is visible. I think so. Is it visible, ma'am?
Yes,
Yes,
it
it
is.
is.
It is
Hello.
very clear.
Yes, sir.
Yes, sir.
Okay. Okay, okay. So yes, for as ma'am have mentioned, the today's topic is really interesting. That is conventional drug designing versus in silico drug designing for the discovery of this COVID-19 medication. So before I start my topic, I want to divide this topic in three categories. That's why I've intentionally written that like the first portion of our presentation is will focus on conventional drug designing, then we'll focus on in silico drug designing, and followed by how this drug design techniques can be useful for the COVID-19 medication. So my presentation part includes several categories. Like I mentioned, first we'll see the what is drug discovery and development. Followed by we'll go for a conventional drug designing. Then we'll come for rational drug designing, which includes in silico drug design, structure-based and ligand-based drug design. And lastly, we'll see the role of in silico method to discover COVID-19 medication. So before starting this topic, very first thing you should know what is drug discovery and development. So many times what we do is we are pronouncing or sentencing this word throughout that drug discovery and development. But these are the two different things that includes two words. One is drug discovery and second one is drug development. Now drug discovery includes your target identification, target validation, lead identification, and lead optimization techniques. And drug development includes majorly two categories, that is preclinical and clinical studies. So remember the thing is these are the two different terms, drug discovery and development. And majorly the in silico methods majorly focus on the first portion of that drug discovery, that is like target identification, validation, lead identification, and lead optimization. First thing is that what is the procedure. So via this pictorial view you will able to understood very easily how drug discovery and development starts. So very first thing, if any kind of a diseases occur, you first thing that you are aware about the identify a disease, why these diseases occur, or what are the reasons for this disease, what are the cause and symptoms and all. So first thing is to identify a disease. Next thing is, once you identify disease, you should isolate the proteins or a biomarkers which are particularly responsible for the disease. Third thing comes is find out a drug or a lead molecules which can be effective. I'm telling can be effective as it is not reached up to the market or FDA approval. So find out a drug or a lead molecule which is effective on a disease. Third thing comes the preclinical testing. Fourth thing comes after a preclinical formulation and scale up. And next thing comes after all thing is go clinical trial and see that that whole that portion requires years of efforts. You can see over here for only isolation of protein you need 2 to 5 years. So if you take a mean of all these things, it requires average 10 to 15 years to develop a one new drug. So it is a very tedious task to develop one drug to reach to the market. So years of efforts are required to develop a one drug molecule to the market. Now next thing is what is the timeline. Now here you can able to understood what are the timeline required for the drug discovery process. Now remember one thing, it is drug discovery process is like finding out a needle in a haystack. It is a not impossible task, but it is a tough task. and it is a very long journey to develop one drug towards the market. So it will see on a diagram, you can see that drug discovery. Say for an example, you have started with 5,000 compound. After the screening, nowadays you should start with billions of compound and screen with 1,000 compound that comes after drug discovery, and for a preclinical only you can see that 250 molecules. And after a clinical, only 5 molecules. And after the clinical trials, maybe chances of one FDA long and risky process to develop one about the 1.2 to 1.4 billion dollar for the time of 12 years. It requires a time. Now we have first thing, conventional drug design. Now conventional drug design the designing which through the whatever the approach which includes three
His
screening
voice is
minus
breaking, I think.
random screening. Hello, am I audible?
Yes, you're
Hello.
audible. Yes.
Okay, okay, there are some background noise are coming, ma'am.
I think everyone should
Just
mute
if
their.
you
Please mute
Yeah,
all
yeah.
the participants. Mute your mic so that it will not
Sir, please unmute your mic, Ashish sir.
Sir, I think all have muted. You can unmute and I think Mr. Ashish has not unmuted.
Yeah.
Can you unmute from your end?
Rakesh, you call him. Call him. Because I
Monica.
don't think
Yeah.
everybody has unmuted. So maybe he's not able to hear. Just call him once.
Mm-hmm.
Yeah, ma'am,
Yes,
I'm
ma'am.
doing it.
राकेश क्या हो गया?
I think some range problem, ma'am.
am I audible now?
Yeah.
Yeah, very much.
Yeah. Now you are audible.
Hello.
But
Yes.
you have to
Yes,
go
Audible
back
now?
to that. You go back to that slide. Because I think it started from the
Okay, okay.
beginning.
Yeah. It has come to the first slide.
Yeah, right.
Are you there, Ashish sir?
Yes, yes.
You can start, sir. You can resume from the slide number 7.
Just one second.
Yeah, now you are visible and your
Yes, sir. You're
slides are off.
visible. Yeah.
Okay, fine. Just give me 1 minute. There's some issues.
क्या हुआ क्या? I don't know what's happened. राकेश क्या प्रॉब्लम हो गया? राकेश
Okay. No, I think Monica, he has muted again. Is this muted? See?
Yeah, his is muted.
Muted. I think someone should call him and.
यार उसको मोनिका अरोरा please call him. Because
So
so
see,
much
you
of
can
time
see.
is going.
Ma'am, I think he
Yeah.
is sorting out the thing.
I think some problem, internet issue with
Yeah.
yours thing also. So I think he's sorting that out, ma'am. That is why he's
Wait, I'm
calling
getting a
again.
call from
Acha, he is
Rakesh.
making some call.
No, actually, what has happened? He has some network issue, and his laptop has stopped responding for a while. So he has just called and said that please ask the participants to just stay there for a while. He'll be back in 2 minutes.
Okay, fine.
Even I got an issue, ma'am. Suddenly my laptop stopped working.
I would like to say, participants, thanks a lot for being with us. There's a little technical glitch. We are waiting for that. Please bear with us, and our apologies for the delay. In the meantime, if you have any queries during the session regarding the lecture, you can put it in your chat box and we will answer the questions. Sir will answer the questions once his lecture gets over at the end of the session. We will give you time. So, whatever queries you have, please go on putting it in the chat box. Also, one request to the participants, kindly fill up the feedback form before you leave the meeting. Thank you for being with us. We will be resuming in a short while. Please bear with us for the technical glitch. Dear participants, we know it's a little embarrassing for us also, but technical faults we can't handle. Be with us. We'll be back shortly. We are sorry for this technical glitch. Kindly bear with us. Sir is already back with us, so we will be starting in a few minutes. In a few minutes, we'll be starting with us. Welcome back, sir.
Yeah, sorry for the interruption. I apologize for this.
Okay, so technical faults cannot anyway. Nobody can handle it.
Yes, yes, no, that's it.
We'll resume, sir. We'll resume.
Yes, yes, sir. Just 1 to 2 minutes will resume.
Yeah, sure.
Please, ma'am, the pop-up shows that host disabled participant screen sharing. Hello.
Sir?
You can,
Yes,
you
sir.
can, sir. 1 second. I will make you. I will allow that.
Yeah.
Yes, sir. Now you can share it.
Okay, okay.
Some background noise is going on. I think we can now tell everybody to mute their mics.
Hello.
Yes, sir.
Yes, my screen is visible now.
Yes, sir. It's visible.
Okay, okay, okay. Thank you. So
Harshit, you
sorry
mute
for.
your mic.
Hello.
It's okay. Yes, sir.
Okay, okay. Shall I start now?
Yes, sir. Please start, sir.
Okay, okay, fine, fine. So sorry for the interruption. We'll quickly we just start with our topic. We have seen that what is drug discovery and development methods. What are the steps that is included in drug discovery and development methods? And we have also seen that how many years it required for the one cycle of a drug. So it will take around 12 to 14 years, as well as the cost will be 1.2 to 1.4 billion dollars. So now we are starting with our first topic, that is conventional drug designing. So conventional means traditional drug designing. Now you can do conventional drug designing on three different approaches. One is serendipity. Serendipity means totally on a base of luck. If you have a luck, then you may success. Now second is like a random screening. So you can see from the pictorial view that see the whatever the sentence is written, someone is from a city from where the terrorist belongs to. So you cannot stop him like that, okay, as he is belonging to the city of terrorists, so we have to detain him. So random screening is like a totally random approach. You just collect thousands of molecules and do synthesizing and do random screening. It's like that. But again, chances of your success is very less. Third thing you can go for is chemical modification. So basically, conventional drug designing is divided in the three broad different categories. One is serendipity, second one is random screening, and third one is chemical modification. Now, first is serendipity. So serendipity, what itself suggests that it is accidental drug discovery. You may not want to discover something, but by accidentally you are discovering something. So that is something known as serendipity. Yes, there are so many kinds of examples are there uh, via which drug is discovered through the serendipity approach. So very well-known example is penicillin, which is discovered by Alexander Fleming. He was just looking for the research on the influenza virus, and by accident he has found out that antibacterial activity of this penicillin mold. Same was like for a meprobamate, that British drug house limited was working on antibacterial, but they are getting the activity like sedative hypnotics. Same was the case for the chlordiazepoxide. They are looking for the dye formation, but they are getting antipsychotic activity. So means to that you are not aiming for something, but you are unfortunately or by accidental you are getting something. That is something known as serendipitous discovery, which is the first type of the conventional drug discovery methods. Now, second thing is that random screening. So random screening means you just collect the thousands of molecules, just synthesize it, and do the biological activity. And if your luck is there, then you may got success or not. But nowadays, this random screening approach is particularly replaced by two methods, that is combinatorial chemistry. I just need to quickly look on what is combinatorial chemistry. So it is the approach which gives you synthesis of 1,000 molecules at a time. So that is something known as a combinatorial chemistry. And second one is high throughput screening. Now, high throughput screening means if you want to take a biological activity of thousands of compounds at a time, then you should go for high throughput screening approach. So random screening can be done with a approach of combinatorial chemistry as well as high throughput screening. But again, here cost is more, chances of success is very less, and you may got failure. Whatever the research you are looking for, or whatever the objectives you are looking for. So this is the second one approach of random screening. Now, third one traditional approach is about a chemical modification. So as the diagram suggests, I have put one example of this chemical modification. Here this drug is cimetidine. So what they have did some changes. Instead of this imidazole ring, they have replaced with a furan ring, and some chemical changes they have did. And what was their aim is they may get activity like a cimetidine, or may more activity as compared to the cimetidine. Now you can see over here also, whenever I'm changing my molecules configuration in the sense, if I'm changing a functional group, then whatever the shape is supposed to the same like the previous drug. So you can see over here reference and new molecule. The shape of the key is same, so that I am able to open the lock. Lock means the receptor. So what was the aim of chemical modification? We are changing certain kinds of functional groups, but the shape of this functional group should remain same. Now, how you can do this chemical modification? Very first thing is homologous approach. Like you can go for the same series, methane, ethane, butane, propane, pentane, hexane, etc. So with the aiming that the same series will give you either additive effect or a same effect. So very first thing with chemical modification that you can do is via homologous approach. Now second thing is molecular disconnection. You just break the molecule, and with the breakage of molecule, whatever you are getting, that have a desirable biological activity. So you can see that via molecular disconnection approach, morphine is converted into the pentazocine and meprobamate. So with it, like same activity they are expecting. Third one approach for this chemical modification is molecular addition approach. Now what you can do in molecular addition, just add some functional groups or some atomic groups towards your molecule and you will get the activity. So if you add it in a morphine, like over here, CH3 is replaced by CH2, CH double bond CH2, and you will get nalorphine. So third one is approach you can do is molecular addition. Fourth one is isosteric replacement. Like you can replace oxygen with sulfur. You can replace amine, say for example, carboxylic acid with sulfonamide group. These are known as bioisosters. I'm not going into in a detail what is bioisosters and all, but bioisosters are like a similar kind of electronic as well as spatial characteristics, and we are aiming for that bioisosters having the same biological activity. So here I have put the example of para-amino of benzoic acid, that is used particular target for the sulfonamides. So what sulfonamides have a common functional group is that COOH group is replaced by this sulfonamide function, SO2NH2 function. So fourth thing you can do is chemical isosteric replacement. So in a chemical modification, in short, four broad approach you can do is one is homologous approach, second one is molecular disconnection approach, third thing is molecular addition, and fourth one is isosteric replacement. So these are the three different steps we have seen of conventional method. First one is accidental or serendipitous, second one is random screening, and third one is chemical modification. Now what is the problem with this three methods? These three methods are really time consuming. You may desire one luck, then cost is also more. You may require more manpower is required. Then chances of success ratio is very less. And especially this kind of a method is not effective in pandemic condition. Like in this COVID situation, we cannot wait for a 5, 10 year or 12 or 14 years to discover one molecule. We need the rapid discovery of the molecule with the effective therapy. So for rapid discovery of molecule with the effective therapy, you may switch over to the approach that is something known as rational drug design. So rational drug design, the part of one rational drug design is in silico drug design. Now in silico means this, the word includes the silicon chip. So computers are generally contains the silicon chip. From that, what is given? So in silico, using computational methods, if you are designing something, then it is known as in silico drug design. So you can see that in silico drug design, it is a term that means computer-aided molecular drug design. In another word, we can say that it is a type of a rational drug design which uses the computer. Various softwares have been used currently for as a in silico drug designing techniques. And what was our aim? Our aim is to do a rapid discovery with a lower cost and with a good results, with good therapeutic efficacy. We are aiming for this, and that's why we are using this in silico drug design techniques. Now, what is the computational or computer-aided drug design? So basically, computer-aided drug design focused on the mathematical equations. Some mathematical equations are given to the software, and they will work on a mathematical approximation, and these mathematical methods are used for a predicting of a drug properties. So computer-aided drug design means the efficient mathematical approximation that can calculate properties of future drug molecules and helping in the process of drug design and discovery. Now, why we need to do this rational drug design, and what is our aim to do that rational drug design? So rational drug, why we are doing this computer drug design? For rational drug design and testing. Second is a speed up screening process, which was previously the drawback of traditional methods or a conventional method. Here we can do even 1,000 or lakh compound per a day with help of the softwares. We are not, remember, we are not consuming manpower, human power we are not consuming. We are not consuming instruments. We are not consuming chemicals. Without this, we came to know that which molecule needs to be synthesized. We can take a decision from 1,000 molecules or lakhs molecules, which molecules to be synthesized or which not to be synthesized. So that results you are getting with the help of software, and this software results are also reliable. So why to not adopt this kind of approach, which saves your time, cost, as well as give you efficient results. So for efficient screening, you can go for this computer-aided drug design. And one thing is fails drug fast. As I have mentioned, if you want to take a decision which not to be synthesized or which to be rejected, so via computational result, you can reject this. So it will ultimately it is saving your cost as well as time of your whatever the process for the research. Now, see, this is the comparison. If I go for a one molecule cost in a US dollar, so for computer modeling, I only require 10 something about 10 US dollar. But when I'm going the next bit much. Now, one more thing is, suppose you have start with a say for example 1 lakh compounds, and with the random screening and all these things, because random screening again requires the synthesis of that compounds. Then after synthesis, you require to biological evaluation. So if you do biological evaluation on a rat study, per compound cost is something about 12,000 USD. So you can assume if your compound is 1 lakh, then how many cost is you are spending on this particular thing. But if you have this technique, computational modeling, so per compound you are only spending 10 USD. So you can assume how much cost have been saved. And for as a pharma company, the first thing is a cost and effective drug development. So to do cost and effective drug development, you must adopt this approach, that is something known as computer-aided drug design or in silico drug design. Now, here you can see the one case study. Now, in a traditional high throughput screening, they have something started with a 4 lakh compounds. And 4 lakh compounds tested, and out of this, they only got 81. So the success ratio is around 0.021%. Now, virtual screening using in silico methods, they have started with something about 2 lakh 35 compounds. After doing the computational techniques, they have not synthesized the compound, they have not tested the compounds. Remember this thing, the computer has told that you just go for the 365 compounds. So without investing cost, computer has told you that just go for up to 2,35,000 compounds. You just go for 365 compounds and see the results. Out of 365, 127 have a IC50 value that is less than 100 micromolar. So you are getting a potted compound, and your chances of success is about 35%. So you can see the direct comparison when I go through the conventional method. Now this example gives you clear-cut idea why one should adopt this in silico drug design techniques. Not only cost concern, as well it will also increase the success ratio. We are not claiming this drug is reached to the market, but see the success ratio is that. Only for with a traditional you are getting only about 0.021 and here 34.8, near about 35. So why to not adopt this approach? Now we are moving towards the computer-aided drug design. So computer-aided drug design, basically it broadly divides into two things. One is you can go for ligand-based drug design. And second one is you can go for a structure-based drug design. When you adopt or ligand-based drug design, when your ligand is known, your basic molecule is known, then you can go for ligand. Around December and January 2020, the proteins related to the novel coronavirus are not reported. So what they have adopted? They have adopted the repurposing of whatever the antiviral drugs are used previously. They have started to give the patients. But we know that success or efficacy of this repurposing drugs are patient to patient vary. Until that, there is no specific therapy for a novel coronavirus related disease. So how you can start with the computer-aided drug design if we are comparing with this COVID-19 situation. So you can do drug designing on a two basis. One is ligand based. Suppose, say for example, you are knowing the structure of remdesivir. So you just do the chemical modification on remdesivir and just go for chemical modification with hoping that it will give a same activity like remdesivir or maybe higher activity like remdesivir. Whatever the result, when you are putting it practically, at that time we will realize, but that is our rationale. Now second one is a structure-based drug design. When your protein is known, now if you go through now in a protein database, so many structures of protein related to COVID-19 are reported. So in that situation, you can go for a structure-based drug design as your protein is known. And then you just check various methods of structure-based drug design. So when your protein is known, you should go for structure-based drug design. If your protein is unknown, then you should go for a ligand-based drug design. Now again, these techniques are divided into some portions. Like ligand-based drug design include 3D structure of structure-based drug design include the 3D structure of target. Now you can do docking as well as fragment-based drug design. If your protein is available, if your target is available, then you may go for these two techniques. And again, in a ligand-based drug design, we have three broader techniques. One is virtual screening, second one is a QSAR, and third thing you can do is pharmacophore modeling. Now we'll see one by one the structure-based and ligand-based drug design. So we are starting with structure-based drug design. So first one is docking. So what is docking? We all have learned that lock and key model. So when the lock is, you may able to open if your key is specific. You cannot open the lock with all the keys. Key should be very specific. So same is like a docking. It follows the lock and key approach. That is the best match between two molecules. One is your macromolecule, that is protein, and one is your drug molecule. If it is matches, then it may give you a desirable biological activity. So that is something known as docking. So here, like our pictorial view also suggests, the best matching or fit between two molecules. That is something known as a docking. Now, second thing is why we should go for a docking. Before starting this, if you see the definition, so docking method is a method which predicts preferred orientation of one molecule as compared to the second. So you can see that one is your macromolecule and second one is your micro, means this is your drug, and how it is bind and what is the binding orientation, what are the interactions which are required for having a good binding. So that you can predict via this docking method. Now why the docking? Why it is needed? Why you need to do the docking studies? So first thing is whether your two molecules will interact with each other or not. Because when you are starting, ultimately, when you are discovering a drug, you have a two major questionary things. One is your drug should reach to the target, and once it reach to the target, it should be bind with the target. So binding is very important. Without binding, you may not get activity, right? So that docking gives you whether your molecule is going to bind or not. If it is binding, then what is amount of binding? What are the interaction they may going to interact with which kind of amino acid residues? So that you can particularly check for from a docking method. So basically, if we want to check interaction between protein and our ligand or drug molecule, then we can do with docking as well as orientation, in which orientation is going to bind. So that should be done with the docking methods. Now there are basically two type of docking method. One is protein-protein docking. So you may dock two proteins because if you go for antibodies and certain protein, which also useful as a medicinal agents. So that for that you can check with the protein-protein dockings. But what we are generally doing, we are not doing flexibility of that molecule. So we keep both molecules are rigid and we check the interaction, how they are going to bind with each other. Now, second type of docking is protein ligand docking. Now, here you have a one protein macromolecule and second one is your, that is ligand. This is small molecule. And this is something is binding cavity where this molecule is going to fit. So when you perform a docking, you have first of all protein, you should have your ligand, and then you should able to perform a docking procedure. So what are the prerequisites or pre-requirements for performing a docking studies? You should aware about your target, which is protein, and that should be downloaded from protein database. And you should have an idea about your ligand, what ligand you are using, and then with certain software, whatever, there are so many kinds of softwares are available with which you can perform a docking method. So here this is a binding cavity, and after a docking you will get result like this. So it fits into the cavity and gives you the orientation as well as binding pattern of your compound. Now these are the application. Suppose if you want to screen the 1,000 molecules at a time with the help of a docking methodology, you can screen within a 1 day. Even lakh molecules with the help of some supercomputers, you can screen within a 1 day. So you can go for a database and you can just screen the database and you may get some molecules that you should go with in vivo and in vitro activity. So for this kind of a purpose, then secondary, as I have mentioned, that if you want to check orientation, then if you want to check the binding pattern, binding pocket, with how, what are the amino acid residues are including, so that you can also go with this docking methodology. Now see that that one case study I have put that they have started with something around 1,69,000 compounds. And what they are looking for, inhibition of anticoagulation and anti-inflammatory activities of SMAC venom. So what they have choose something around 1,69,000 compounds. First of all, they do pharmacophore-based screening, that may be ADME or other structural feature techniques that will going to look in a ligand-based drug design. So after a pharmacophore-based screening, they are getting a 300 compounds, and after docking, they are only getting 32 compounds. So see how much screening only you have did with the computation. Now that 32 compounds, you may go for a synthesis and then for a biological activity. So if you can compare, if you don't adopt this kind of approach, you may have to start with a synthesis of this much compound. You have to do synthesis, you have to do biological activity. So how you can assume it requires so many time as well as cost as well as the chemicals and all these things. So that's why these techniques are really necessary nowadays. Now, second one is a de novo approach of drug design. Remember thing, we are looking for a structure-based drug design in which we have two different methods. One is docking that we have already seen, and second thing is a de novo drug design. It is like a stepwise drug design. You may get a hear from. So what I will do in a de novo drug design, first of all, I will identify my binding pocket. Like, so say for an example, this is my binding pocket. And step by step, I am including the functional group. Suppose, say for an example, just assume like that, particularly this portion. Say for an example, it is hydrophobic in nature. So for a hydrophobic, you require a hydrophobic group in your drug. So I'll put hydrophobic groups in this particular portion. And suppose this portion is hydrophilic. So I'll put on a second way the hydrophilic groups. What my aim is, I sequentially build up my molecule and I end up with something that is have a better binding affinity with the target. Now, in which condition you should go for fragment based drug design? Suppose you have a compounds, but out of this compound or a drugs, any compounds are not effective. You have checked all the things, but it is not effective. Now, next your task is go for this fragment based drug design. In case of you have a inavailability of existing drugs to treat your disease, then you can go for a this fragment based drug design, but it requires the protein structure should be known. Without protein structure, you may not go with this fragment based drug design. So you can go sequentially build up and you are end up with one molecule which have a good binding affinity. So this is something known as fragment based drug design, and these are the some steps like growing, linking, then lattice based sampling, and then molecular dynamic based methods. So these are the things that you can do with a fragment based drug design. Now we end up with a structural based design. We have seen in that I'm very going superficially because time constraint is there, and to know this method in a depth, it requires hours of time. I can discuss endlessly if you want, but I'm going to make it simple so that you can able to understood, because many of people are not aware about this. So my aim of today's lecture is you may have idea what is in silico drug design, what are the techniques involved in silico drug design, and how we can use this in silico drug design techniques for a COVID-19 medication. So we enter into the ligand based drug design. Very first thing we look for the QSAR. Now, to remember QSAR, that is a quantitative structure activity relationship. So you may have a compound and they are reported for the biological activity. And you just do the mathematical approximations. You just develop one equation in a simple way. If you learn what is QSR, I just give you a very simple example. Suppose, say for an example, you want to make one good cricket team. All of you are might aware about the cricket team. So what will be your combination if I am choosing my cricket team? My combination is like I'll choose 4 to 5 bowlers. I'll choose 4 to 5 batsmen and at least 1 to 2 all-rounders. This will be my probable combination when I am choosing, say for example, good cricket team. So how I got knowledge that 4 to 5 bowlers are required, 4 to 5 regular batsmen are required, and 1 to 2 genuine all-rounder is required. From where I can get knowledge? I just get knowledge from the previous data, from previous matches. I came to know that this combination must require. So this is something like that. From a previous compounds, I have some knowledge. Yes, these are compound better effective. I just need to put this combination. And I am designing my compound on this way. So I have a previous compound with biological activity. And what I am doing is new compound with improved biological activity. And this thing will be happen. You can get activity approx activity or mathematical activity of your compound with the help of QSAR. It will give you, if you are designing a new compound based on a previous compounds, it will give you the probable biological activity of your compound. So based on this, you can decide this molecule needs to be synthesized or not. So this is something known as QSAR. So as I have already mentioned, that is a quantitative structure activity relationship. So that requires to develop a new molecule. So what are the requirements for a development of a QSAR is you should have a, first of all, begin with a biological activity, and then you can go for a molecular descriptor. Descriptor is just like that all-rounder, regular batsman and bowler. So same way in case of if you correlate with the chemical structures or a functional groups. So various functional groups are my all-rounder as well as bowler as well as good fielder, anything. So I'll choose the molecular descriptor. Then what are these molecular descriptors? So like electronics and spectroscopic or other properties I have to consider. Electronic or spectral features I have to consider when I'm choosing my molecule. Then what I will do is I have a physiochemical parameters and biological activity. So what I will do that I will correlate this physiochemical parameter with biological activity. And I am assuming that the biological activity of my compounds is due to the physiochemical property of my compounds. So that physiochemical properties may going to play a role in a biological activity. And that is my aim. That was I'm assuming. And I'm deciding which physiochemical property is important and which is not important. So whatever the property is important, I'll put on my chemical structure, and then I will predict the biological activity of my structure. So biological activity here you can see that is equal to the physiochemical properties. So your assume like that your biological properties totally depends on a physiochemical property, and then you may end up with some equation which gives you the probable biological activity. So this is the probable way to perform a QSAR. You should have a compound library with biological activity. You should have a set of compound, then activity data. You should have a molecular descriptors, and then you just develop an equation. Y is equals to f of x, x is a physiochemical parameters. And you just predict and interpreted the biological activity of your future molecules. So that is something known as QSAR. Now, second thing is virtual screening. Now, virtual screening means if you want to screen thousands of compounds at a time. So screening of 1,000 compounds at a time that you can do with a virtual screening. So for that, based on only you can say that drug-like properties. We are not considering that structural-based drug design. So for a ligand-based drug design, you can do screening on the basis of ADMET profile. You can predict. There are so many kinds of software as well as online tools are available, which can predict your ADMET profile of your compound. And based on ADMET profile, you just decide. If it is passed to the ADMET profile, then you can move ahead for further. So that is something known as virtual screening. Now, how you can do screening? One, as I already told you, based on ADMET profile. One is one base of structural queries. So structural queries can be anything like a 2D structural queries and 3D. Now, 2D is the first pictorial view is about 2D. Now my query is like this. I have a benzomerazole and this benzamide moiety. So if I put a query, I'm getting a result near by whatever the database you are looking for. You just put a query on that database and it will give you result based on your query. Remember this one more thing, 2D similarity means not like an exact structure match. They will give you result on a based of physiochemical parameters matching. Means like log p value of this compound. Say for an example, log p value is 2. So you'll get the compound which have a log p value nearby 2. They will give you all the results of compounds which have a log p value is nearby 2. So this is a 2D. Now second you can do is 3D substructure searching. So in case of this 3D structure, 3D properties are involved. So you can see now here the like hydrogen bond donors, acceptors, then lipophilic characteristic, electronic features, spatial features. So you can just I just give you one example. Suppose if you are giving for a zinc database, just go through zinc database. You have an option to screen your compound on based on property. So whatever the properties you require, you just select that property and gives its value. Say for an example, I require hydrogen bond donors between 1 to 5. So that gives you results of only the compound which have a hydrogen bond donors is in between 1 to 5. So based on 3D similarity, you can go for screening of compounds. And third one is a pharmacophore approach. So pharmacophore means 3D representation of your molecules. That is something known as pharmacophore. If you are considering a 3D representation and 3D parameters which is involved in a biological activity, then this is something known as pharmacophoric approach. Now, what are the pharmacophore features like hydrogen bond donor, acceptors, hydrophobic, hydrophilic, positive charge, negative charge? These are the pharmacophoric features. So based on the pharmacophore, you may also screen the compound. And this is the based on your ligand only. So that's why it is included in a ligand-based drug design. Now this how you can develop a pharmacophore. So this is my representative molecule, and based on I am getting this results. So first of all, you have to choose first your standard or reference molecule and give its definition, like how many hydrogen bond donors are there, how many hydrogen bond acceptors are there. So based on your query, it will gives you the nearby pharmacophore. So this is how you can develop or find out different pharmacophore which is near to this strata, right? Now, what are the advantages of CADD? So I'm just briefly, I have switched over to the in silico drug design. Again, I'm just giving in 1 minute recap. It is divided into the two things: structure based and ligand based drug design. Structure based design, we have seen two techniques: docking, it is matched between two molecules, and fragment, it is sequential building. Then we'll switch over to the ligand based drug design. In that, we have seen that QSAR, then virtual screening and pharmacophoric approach. Now, what is the aim of this computer-aided drug design? Is to save your time, cost, to give accurate result, then information about disease. So these are the application which is or advantages which is involved with the computer-aided drug design. Now, these are the various softwares via which you can perform different kinds of method. Suppose for chemical representation, you can go for a CAMP or Marvin stage. For molecular modeling, you can go for CHAM, GROMEX, etc. softwares. So this is slide is particularly gives you the idea for which kind of techniques, which are the softwares are available. Suppose if you want to go docking. So whatever the example I have put over here, these are the free softwares. Just go and play with the softwares and you can learn the softwares. And if you have any queries regarding this, and I may help you in this also. So for docking, these are the free softwares which are available. Same for the binding site prediction, target prediction. These are the Swiss target prediction. This is an online tool which is freely available. For QSAR, this is a paid, but build QSAR is again free software that you can do for a QSAR, performing the QSAR techniques. Remember, I put just few examples. There are so many kinds of softwares are available, but I'm just putting some of examples so that you might get idea what are the softwares I can use if I am beginner. Then what are the softwares I use for a particular kind of a technique. Suppose if you want to do ADMA, then you can this before from Schrodinger. Again, it is paid basis, and here CZM is free basis. This is an online tool. So if you want to screen your ADMA property, then you may just go for CZM if you have not access of Schrodinger. So you can go for CZM. So I put up both example, paid and unpaid. So mostly as academic institute, you may not have a grant for the paid software. So you can play with the whatever the software which are freely available. As I have mentioned, that docking for Autodoc and the SwissDoc, both are freely available. So you can go for this. Now these are the some examples which the drugs are discovered through these approaches. Like norfloxacin, it is discovered with QSAR. Then donazepam, again for docking and shape analysis. Based on this, it has been discovered. Then indinavir, it is come for structural based drug design. Then sequenavir, Denovo drug design. So that we have already seen these techniques. So you now able to realize that if this techniques give me promising results, and based on these techniques, I can discover a new molecule with a better biological activity. Now next, our third portion of my lecture is role of in silico method for discovery of COVID-19 medication. So we have seen, now you have an idea about what are the in silico methods, what are the types of in silico methods, and when and where you can apply the in silico methods. So now we are applying this knowledge towards the COVID-19 medication. So for a COVID-19 medication, you can use broadly two techniques, bioinformatics and chemoinformatics. Remember, this is a drug design, in silico drug design techniques. So bioinformatics is related to the biological target. If you want to work on a discovery of target identification and validation, you may have a knowledge about bioinformatics. And if you want to discover a new molecule based on their properties, then you can go for a chemoinformatics. So broadly, bioinformatics is about your target identification and validation process, and chemoinformatics gives you a lead compound. The third one step of drug discovery, lead identification as well optimization process. You start with the chemoinformatics. So for a COVID-19 discovery of COVID-19 medication, you can use both of approaches together, chemoinformatics as well as bioinformatics. So here you can see that this role are parallel. Both knowledge you have to use. You should go for gene or protein. This is bioinformatics, and this is your chemoinformatics. So both approaches are applied to discover a new medication for COVID-19. Now, how this role of bioinformatics are applied? So see, as I have mentioned, it is particularly, I'm not going in a detail, but just to do superficially, it is useful for target identification and validation. So for target identification, you need to do different steps. Now, what is your target pattern? What is your gene? What is your classification? What category it belongs to? Whether it is ion channel or whether it is a G protein coupled receptor. So you need to identify your target classification. And then you come up with a biomarkers. Which is a biomarker? So for an example, if we are talking about the cancer disease, so tyrosine kinase is one of the biomarkers. So you should have idea about tyrosine kinase structure, what is tyrosine kinase gene, and why it is responsible for the cancer disease. So target identification as well as target validation, you can do with the help of this bioinformatics. So these are the methods, different kinds of methods. Remember, to learn this individual method, it requires again a months of time to learn one method of this bioinformatics. But just you should have idea what you should follow when you start with these bioinformatics methods. Now these are the bioinformatics tools where you can. These are all our online tools. Suppose if you have a gene sequence for some disease, so what you should do is just you put query of gene sequence on this particular databases. So that query gives you nearby results. Suppose if you are looking for, say for an example, TNF alpha. So you should have some gene sequence of TNF alpha. You just put on query. And for COVID-19, if we correlate, so most of the target of COVID-19 are related to the MERS code, which was previously occurred. So 80 to 90% are structure similarity. So that very results from this database will get. So your work time has been reduced. You just need to identify only sequence. Just put on this target database sequence of your gene, and they will give you what the nearby structure of protein is. So within some time, even in a minute, or if your protein is longer, in few hours you will get what is your nearby target. So you are predicting your nearby target. and whatever the target are discovered for COVID-19. So most of this database have been used because previous SARS-CoV, this is SARS-CoV-2. So previously it was SARS-CoV. So that gene sequence, that targets were recorded. So based on this, we have discovered newer target for the COVID-19. Now, if you want to do research, particularly based on in silico drug design, so before entering into this, I just want to suggest, if you want to do research in silico drug design, first you should have a idea of your disease thoroughly. Idea in the terms of disease means you should aware about what are the targets of your disease, and out of that target, which target probably going to play important role for the disease. So if you want to say for or do research via performing docking and IDMT studies, so first thing hurdle that comes with you is how to choose the target. So for a target choose, you should go for a protein data bank, just put a query. And just in a 1 minute, I'll brief out this thing. Suppose if you are looking for to work for the COVID-19, say for example, main protease, that is 3CL-Pro and M-Pro. So you just go to the protein database. It is a universal database for a protein where crystallographic structures are reported. So you just put a query, you will get the results. And try to choose the PDB IDs which have a X-ray resolution greater than 2 angstrom. Why you are choosing this? Because this resolution structures are more authentic as compared to the less resolution. So if I want to do research on COVID-19 and my part is start with the in silico drug design, so I know about the structure of whatever the targets of this COVID-19. So COVID-19 target is divided into two things. One is related to the virus and one is related to the host. So for related to the virus, we have we can say that certain kinds of proteins, like efficient main proteins are spike protein as well as nucleocapsid proteins. Apart from this, we have 16 different kinds of non-structural proteins. They are also going to play important role. So these are the target till date reported for the COVID-19. So if you want to do research, you may choose the area of your target or interest of your target, and then you can move ahead. And I have put the examples. You can check all these examples. These targets are already available in protein database. So you can download this protein structure from this, you go for a docking studies, and then you may end up with something compound which have a good binding affinity. So these are the particularly virus targets are available. Now out of this, spike protein, nucleocapsid protein, then 3CL-Pro, M-Pro, these are the main protease. Then PL-Pro and RDRP, these are can be considered as the main targets. I'm not ignoring this kind of a targets, but these are the main targets. Why I am sharing with you? Because I am also working with my 4 papers are on a pipeline for this COVID-19. So what we have did is we have choose this kind of a target. Then we have choose the database. There are so many kinds of a database available. And first of all, we did the ADME screening based on Lipinski rule of 5. Now, what is Lipinski rule of 5? That your molecular weight of your compound should not be greater than 500. Then hydrogen bond donor limit, it should be 5 or less than 5. For hydrogen bond acceptor, it should be 10 or less than 10. So if molecule have this kind of a property, then your compound may have oral activity. So for oral activity of compound, the Lipinski rule of 5 is very important. So what we have did, we have screened our compound based on Lipinski rule of 5, and then on the basis of screening, suppose in a one paper, we have started with 300 compounds. Then we end up with ADME screening based on Lipinski rule of 5. We got something around 17 compounds, 70 compounds, and then we did a docking. In a docking, when you're performing a docking, you should have one standard with you. Standard can be drug or whatever the ligand present in your protein structure. So as a standard in one paper, we have taken a remdesivir. Suppose your remdesivir docking score say something around -8. So whatever the compound, we are getting greater than less than -8. Suppose we are getting score -9, -10. This compound we have screened. So we have taken 300 compounds that I'm just sharing one of our research paper in which we have did like this. That 300 compounds we have started. Then based on NADM, we are getting 70 compounds. And out of 70 compounds, based on docking score, we have seen 5 compounds. And then we have checked toxicity. So after the toxicity, we got 2 compounds which are safer in a toxicity profile also. So you can assume that I have started with 3 compounds and I end up with only 2 compounds. And I did with only this computational techniques. So that 2 compounds now you can directly go for in vitro or in vivo studies. So this is called as a basic pioneer genuine research that you start with this. So for this, you need to knowledge about these targets. So these are the target which is already available in the protein database, and these are the virus target. Now these are the human targets. Now suppose spike protein bind with this SE2 human domain. So these are the human sites. So approximately 7 or 8 human sites are there which favors the growth of virus. So you can also inhibit this particularly this site so that virus binding is not possible or virus growth is not possible. So you may work on either directly on virus target or you may work on a host-based target. This is a human target. If you can inhibit this target, maybe virus growth will not occur. And some of this already reported in a protein database. You just go through this and you will find out the protein structure in this protein database. And some of the example I have also put this. So these are the some targets which is related to the human. So remember the thing, as now structure are available, it is my advice is that you just first start with the ADME properties, then you can go for a docking studies and then you can optimize your molecules. Now, these are the some drugs which are used to treat this COVID-19 infection. So, like remdesivir, the target is RNA polymerase. Then lopinavir and ritonavir, the target is 3-CL protease, which I have already mentioned previously. Then for a disulfiram, the target is peptide-like protease, that is PL protease. So, what you can do is for a docking, you can, if you want to say for an example, work on RNA polymerase target, you just take a remdesivir as a standard, whatever the results of docking score, less docking score you are getting as compared to the remdesivir, you may go ahead for this. So this kind of research flow you can follow to do research in particularly related to the COVID-19 pandemic. Now, this was the role of bioinformatics. Now, second importance is a role of chemoinformatics. So, role of chemoinformatics is like you can screen your compound from the database. Then you can, as I have mentioned previously, you can do for prediction of physiochemical and biological properties like ADME and toxicity properties, and then you may screen the compounds. So, chemoinformatics is also important. What you can do with chemoinformatics, that you will get screening of chemical database. And remember the thing, both approach should be parallel. Like bioinformatics and chemoinformatics must require for efficient lead optimization technique. So in a chemoinformatics, you can go for screening of chemical database, then natural product database. So many kinds of database are online available. Some of are free, some of are paid. Then screening of specific plant constituents. You may go with specific plant, choose the phytoconstituent available in that plant and do for docking or ADME or anything you can do. And based on this, you may end up with something, a novel compound, which have a good binding affinity in silico. Then you can also screen FDF drug. So these are the some approaches you can do. Suppose you are thinking about to do research in a COVID-19, then these are the some clues that you can do via if you want to do research in silico methods. Now these are the example of small molecule database, synthetic molecules, small molecules like drug bank, zinc, then NCI open database, Campbell. So from that you can take millions of compounds. So zinc particularly database have billions of compounds. So you can get billions of compounds and then you can screen it based on your whatever the configuration of your software. You can do this procedure. So these are the examples of a molecular database from where you can get the library of your compound. Because now in a COVID-19, our first aim is repurposing of drugs or repurposing of chemicals. So we are want to identify from whatever existing molecules which may be more effective. So that is our first approach. Because if we go for a like a conventional drug designing, it will require a time. So that's why we are doing this. So these are some database that you can do. Then role of natural products. Remember the thing, natural products gives you very promising results with a lesser side effects. So you can use natural products to, for a COVID-19 medication also. So how you can choose natural products? There are certain natural product databases are available that you can screen, or else you can choose particular plant which is reported, say for an example, a good antiviral activity. So you can do hypothesis as it is reported in antiviral activity. I can check on COVID target. You just select any one of COVID target and you just do docking and ADME filtration and all. So you may end up with good results or the compound will have a better results in terms of in silico. So natural products are also going to play important role, and this the workflow is followed. So first of all, you should have a medicinal plant that you go for a pre-treatment like a ADME prediction. Then you can go for a virtual screening. Virtual screening may be based on pharmacophore like ADME properties, safe similarity, and docking methods. So these are the methods you can adopt for a virtual screening, and then you can after a virtual screening you can do a target analysis. So if you are going through a natural products, this may be probably your good workflow for the research. Now these are the examples of natural product database which are available. Some of are the freely available and some of are paid. So these are the few examples. There are so many kinds of database are available, but I have put which are well-known database. So you can go to this database and screen the molecule from the database. Now we see 1, 2 to 3 case study. Actually, what the people have did. So this is a snapshot of people, the researcher which have did the drug repurposing of coronavirus in silico screening of known drugs against coronavirus 3-CL hydrolyzed protease enzyme. So 3-CL main protease they have target with which known drugs. Now see how they are discovering. This is the work. So they have what they have did zinc database. They have started with around 30,000 molecules and screened first of all 4,500 molecules based on their ADME property. Then what they have did structure-based virtual screening based on docking. So whatever the compound which have a binding affinity greater than -8, like -7, -6 is greater than -8, they have rejected. And whatever have a less than or equal to -8, they have accepted. So they have started with the 30,000 compounds. They have did ADME studies, 4,500 compound they got. Then they have did this docking study. And then after a docking study, again, they have filtered on a basis of ADME profile and see they end up with 4 hits for this particular target, 6LU7 and TNSS2. So you can assume they have started with 30,000 compounds, end up with only 4 compounds. And that they are doing with only help of computational methods or in silico methods. And just for a 1 minute, just assume that we don't have this kind of a methods. Then what you will do? You will synthesize or purchase these 30,000 compounds. Then you do biological activity of 30,000 compounds, and you may not get succeed. Out of 30, you may not get even 4 or 5. That will be the chances of your results. And for this testing of 30,000 compounds requires so much time. Again, cost is more, but with help of the software, we are doing this. So this is the one paper. Now, second is of drug bank. Now, what the research group did that in silico repurposing of particular drug bank database. Now, drug bank is a database which have reported the FDA approved drugs. Whatever the FDA approved drugs are that reported in drug database, so they have used around 10,000 compounds, and they have did screening. The screening is based on molecular docking. You can see that molecular docking calculation, and they got out of this 10,000 35 compounds. And finally, they are reporting two things. That is, Fabry state is useful, particular S and Pro. And this molecule, DB02, that is Fabry state, they have reported as a good binding affinity. They have did molecular dynamic studies, ADME filtrations, as well as the molecular docking studies. So they have started with 10,000 around compound, and they end up with this much one, that DB2388, Fabry state, and darunavir. They end up with only 3 compounds that should be explored as the whatever the target they have used. So see, this you can do with, this is a pioneer first part of your research. From this you can go ahead. So why I put this case study is just you can realize what researchers are doing actually. So whatever the method 3, so this is also good journal. So this work is adopted nowadays and being the protein, and then they do in silico study like docking analysis, and they have ended up with only these 4 or 5 molecules. So you may do research on this and you can publish a paper also. And as a pioneer basic research, if you want to do discovery of molecules, so you should start with this in silico, and whatever the results you are getting in silico, that you may go for in vitro in vivo studies. And this is example of African medicinal plant database. So from database, they contain around molecule, this African database. And they contain so many kinds of alkaloids and terpenoids compound. Remember, just see the title. So they have screened alkaloids and terpenoids from African medicinal plants. So this is a database. From there, they have screened alkaloids and terpenoids. So here you can say that 62 alkaloids and terpenoids from 100 plants they have chosen for a target 3Cl Pro, right? And they have ended up with this compound. These are some compounds that they have ended up with. So you can use natural product database, you can use specific natural product plant, you can use synthetic small molecule database for your discovery of COVID-19. So that's why I put this case study. Now my conclusive remark, if you want to do really research in a COVID-19 medication, so first thing you may have idea about the targets of COVID-19. From the target of COVID-19, you choose your target. Then secondary, you choose your molecules. Where you want to do research with small molecules, or you want to do research with some medicinal plants. Then after choosing this, you just screen the molecules based on ADME properties, and then go for a docking studies. And the molecules which are good at docking as well as ADME filtration, you go for in vitro and in vivo studies. And results are really reliable. You can work on this. There are so many evidence are there from in silico molecules are optimized, they're actually giving good result. So that you can do. I just in a 1 minute I share my work history also. So for my cancer work, I have developed some novel molecules regarding a cancer. So I have adopted same approach. I have first designed 100 molecules. I end up with in silico only 25 molecules. From 25 molecules, I am able to synthesize 18 molecules. And from 18, I am getting 8 potent compounds which have a IC50 value lesser than 50 micromolar. So you can assume that how it is reliable. Computer had given me 18 compounds, and actually in a practical, out of 18, I am getting 8 compounds. So my result success ratio is nearby 40%. So why to not adopt this approach? So why this in silico techniques are really important. You should learn. It is really interesting that you can go for this kind of a techniques. Now these are the reference that I have taken from where I have taken case studies and all these things. Now this is things we have a drug discovery lab at our Department of Pharmacy, which is established in 2018. We are providing this software related facilities. We are also giving guidance for the research papers and project work, and we have access of all this kind of a technique in our drug discovery lab. And these are some papers which is from Sumandip only. I have not put the papers which is based on other institute, but this is from Sumandip. We have able to publish this paper based on only in silico studies. They are in very reputed journals. So these are some papers from our department. Now, if you need my help regarding this training of various softwares, or if you want to do project on in silico based, if you want to do research paper writing based on in silico method, I will help you. I'm ready to help you. These are my contact details. You may contact via email, via WhatsApp, anything you can contact. I'll sure help you. Not only for a COVID-19 concern research, if you want to do research on some other areas related to the in silico, you need to do training, then I'll provide it. I'll ready to help you. You can take advantage of this. And lastly, thank you very much to the all, and thank you very much Alamin College of Pharmacy. Thank you Principal Madam for inviting me. I hope you get some newer message after this my presentation. And if you any participant have any question, now session is open to all. Madam.
Thank you, sir. Thank you, Ashish sir, for such an informative and interesting one. Though we had an initial technical glitch, but after that, everything went on very smoothly. Thank you for such great explanations. Okay. And I would request all our participants to pour in their questions in the chat box. One more request, I know I have told already, but please kindly fill in the feedback form before leaving the meeting. Over to you, Monica Arora, madam, for the question, answer, and interactive session. Over to you, Monica.
Thank you so much, Tanushree. Thank you so much, sir, for the knowledgeable insight. The session was really, really very nice. It was very knowledgeable. So we have got few questions. So the first question is from Ankita. So the question goes like that: how drug discovery can be done with automated high throughput in screening? I think this was in the initial slides.
Okay.
I'm repeating the question again.
Okay. She means to know that how high throughput screening is utilized.
Yes,
right,
yes,
ma'am?
yes.
Okay. So for high throughput screening, you have a certain instruments. You use certain instrument which can give you pharmacological action. Suppose if you have a 10,000 of molecules, you just put some micromolar quantities of your 10,000 molecule into the chips. That chips have a message of biological targets. Suppose if you want to do research on tyrosine kinase related to cancer, I'm just giving an example so that you can able to understand. So just take a tyrosine kinase chip which is available in the market that can detect thousands of molecules at a time. You just put your compound into the chips. So they will do real-time working. Exactly whatever happens in our body, they will bind like that and they will give you activity. So high throughput screening is you can say that it is a type of a method where you can get pharmacological activity of 1,000 compounds at a time. But you need instrument for this. You need not software for this. For a high throughput screening, you need instrument. These are available, but this instrument requires really higher cost. So academic institute cannot afford, but pharma companies have. So it is like doing a pharmacological activity of your molecule, actual pharmacological activity.
Hope it is clear to Ankita now.
Yes.
So now one more question has come in the chat box. So it is from the name of the participant is Akash. So the question goes like that: how homolog approach is different from the molecular disconnection?
See, in a homolog approach, what you are doing is you just go with the same series. suppose if you are starting with substitution with same series of alkanes, say for an example, so you go for like methane, ethane, propane, butane. So these are considered as homologous. While what we are doing in molecular disconnection, we are breaking the molecules. And via breaking of molecule, we aim to desire same biological activity. So that is a basic difference. While in a homologous series, no breaking of molecule is occur, only extension of molecule is occur. While in case of this, the breaking of molecule is occur in a molecular disconnection approach. Hope you will find out it is useful. You may get the message.
So hope it is clear to Akash. One more question has come across, sir. So the question goes like that. In virtual screening, how do you get to know that how much percentage of the drug discovered will be successful? Of course, we talk in the term of the percentage.
Yes. It's like that. Whatever the example I have put, that is published in a research paper, and the whatever the results that I have put, that is the actual result. So that's why in the last I have told you my experience. So again, I'm just repeating my experience. I cannot predict how 40% I will succeed, but I am just hoping that the chances of my success is more as compared to the other techniques. So you cannot decide your percentage is maximum like 40% you will get or 30% you will get. The chances of success ratio again is based on whatever target you are choosing, then accuracy of that target, accuracy of your in silico methods, and then after accuracy of your in vitro in vivo results. So based on this, so for in silico results, there is nothing like boundary. Your chances of success is 40-30%. It's not like a boundary. That is the first thing that I want to clear. And second thing is, you may not to he or she wants to know about why these chances of success ratio is more. So that one thing is that assume like that whatever the thing occurs in our body, that is exactly applies or given command to the softwares. So you can play like this if you want to work on a safer stomach. So you may give a command to the software that pH should be between 1 to 2. And you can see how your molecule work on pH 1 to 2. So results are reliable, that's why. And that's why chances of success is more.
Okay. Thank you so much, sir. Hope this is clear. This is clear to Mr. Mahesh because that was the name of the participant. One more question is dropped in. So the question goes like that. In QSAR, how biological activity is related to physiochemical properties?
Okay, that is a good question. So I have given particular example of that's why the cricket team. So suppose if you want to play a cricket match tomorrow, so what will be your thinking? Which will be the playing 11? You will put like 4 bowlers or 5 bowlers, 4 or 5 batsmen, and 1 all rounder. And why you are putting this? That is based on your previous data. Like previous data, previous match statistics you have. So same way like compound, which is first thing is if you want to perform a QSAR, you should have a compound which are already reported for the biological activity. Without this, you may not proceed for the QSAR. So compound which are reported for the biological activity, the first assumption in QSAR is that biological activity is due to the physiochemical parameters. Say for an example, drug binds with a target. A target binding can be hydrogen or hydrophobic binding. So how will you get hydrogen binding? For that you need a OH containing, H containing group. And for if you have that hydrogen donor acceptor group, then you will able to bind with a hydrogen bonding. If you have a hydrophobic group like benzene and etc. things, you have a hydrophobic interactions. So why this hydrophilic and hydrophobic interaction is occurred? It is due to their physiochemical properties. Without physiochemical properties, drug not going to work. And there is assumption is that for if you want to perform a QSAR, that you first assume that physiochemical property is responsible for the biological activity. So that's why I have given this example. Hope it is useful to you. And if you are still have a query, you may write an email to me, or you can have a discussion with me also.
So hope it is clear to the participant named Bhumi. So anybody from the participant want to ask any question can ask. Okay, now it is over to Tanushree. Thanks, sir, for the beautiful interactive session.
Thank you. I hope participants will get a newer message from this.
Yes, yes, it was a beautiful insight, sir. It was, I mean,
And thanks
very
for
interactive
inviting
session
me.
as well as.
And I'll thankful to your management, principal madam, and all staffs for their cooperation. And again, I apologize whatever the difficulty occurred in the starting. Like this.
No,
And
sir,
again,
those
thank you
such
very much.
glitches, it's unavoidable, sir.
Yes.
Over to Tanushree.
Sorry. Thank you, Dr. Arora, and thank you, Mr. Ashish, for the interactive session. I would now request Kalpana, Mrs. Kalpana Devi, to give the vote of thanks and summarize the session in a short while. Over to you, Kalpana.
Thank you, Tanushree. Am I audible?
Yeah, you are audible.
Okay. Thank you, Tanushree. Indeed, today we had a very informative and useful talk. So we learned a great deal about in silico methods and how we compare the conventional drug design with in silico methods. This is a very heated topic. It is there in the MPharm syllabus also. What are the steps involved, that how we can reduce our time in designing during this pandemic world when we require the drugs in such a short time. Thank you, sir, for highlighting this. So the little bit I want to summarize what has been discussed today is, now he stressed upon conventional drug design. First he told about sir told about conventional drug design, that how we can design, how initially drugs used to design that with the help of serendipity. As you know, serendipity is nothing but luck by chance, you can say. And then by luckily, like how penicillin was discovered, and then we have a random screening also. Then little bit it came like chemical modifications, wherein it is done through homological approach, molecular disconnections, molecular addition, isosteric replacement. These are the some of the methods. And then through random screening, like designing or like synthesizing lot of compounds at a time, which is now called combinatorial chemistry or high throughput screening. But there are a lot of limitations which sir has stressed upon, because you know, everyone knows it is a very time taking, a lot of expenditure is involved, and success ratio is also less. So slowly they started for rational drug design. So sir has given a very good detailed presentation on how to go step by step also in rational drug design. So one of them, which is this in silico drug design. So we have seen here divided into structure based design and ligand based design. So in structure based design, he has said about docking, and then fragment based design, and in the ligand based he spoke about virtual screening, QSAR, and pharmacophore modeling. So we saw a detailed study on how to do docking, what are the application of docking, types of docking, what are the steps involved in docking. And then we also saw how we gave an example of anticoagulant, anti-inflammatory activity of snake venom, where we saw that 1,69,000 compounds were screened, and they came up with 326. So it is a very huge success, like screening so many compounds and arriving at very less. And of course, these few compounds can be synthesized in the lab. Then we have Denovo drug design, and then if we have the protein structure is known, then we can go for the fragment based design. Then he also told about the ligand based drug design, wherein we now have about QSAR. He told about how QSAR is performed. Very good example he showed about the cricket team, which actually will never forget how to design, how to go about in a QSR. Then how virtual, he also told about virtual screening, where thousands of compounds can be screened through ADMAT, applying Lipinski rule, 2D substructure cells, 3D substructures. And then he told about focused on about pharmacophore approach, which is totally based upon ligand. Then what are the advantages? He stressed upon the advantages of CADD, like what is the time is required, cost is required, accurate, informative. He has also discussed his own experience that he also screened for antiviral drug. He screened some compounds and then he actually arrived at some, I think 8 compounds, if I remember, 8 compounds, and then he went on the synthesis. So actually you reduce the time and then you really come out with some fruitful compounds which really have medicinal value. And this one, and then coming to the third part, which is like role of this in silico methods in today's pandemic or COVID-19 medications, is a combination of bioinformatics and chem informatics tools can really help in finding out the medications for COVID-19. So in bioinformatics, we had to know the target identification and target validations, for which various online database are available, and we can go with the nearby targets like remdesivir and all, which are known drugs, clopidogrel. And we sir also said about really that PDB IDs were very useful, sir, for COVID-19 targets and for the human host. Really the targets were useful, so now we can work upon it. And then chem informatics, like various approaches like screening of chemical database, screening of natural products, screening of some specific plants and FDA approved drugs. And the data which sir has listed out for the creation of library is Zinc, NCI, PubChem, to name a few. So really it was a very useful information, and we'll try to apply all these techniques. And thank you, sir. With this, I would like to formally present the vote of thanks. Hope I could cover all the points in summarize it. So with this, I would like to present formally the vote of thanks. An ignited mind is the most powerful weapon on the earth, above the earth, and under the earth. And this is possible only through education. Education is the most profitable investment for the near future. On behalf of management, Al-Amin College of Pharmacy, principal, staff, I take this opportunity to thank Mr. Ashish Pisa for delivering his valuable speech. We'd like to hear from you again in the future, sir. Thank you so much, sir. At the very onset, I would like to thank our convener, Dr. Shobha Rani Hiramed, Madam, Principal, Al-Amin College of Pharmacy, who is also the Dean at RGHS Pharmacy, Bangalore. Without her guidance and motivation, we would not have been able to organize such a wonderful and fruitful event. Thank you, ma'am. I would also wish to thank the 2 webinar organizers, Dr. Preeti Kawa, Madam, and Mr. Rakesh Panda, for the capable way in which they have organized this webinar series. My heartfelt thanks goes to our beloved HOD of Pharmaceutical Chemistry Department, Dr. Monica Kachro, Madam, who believed and gave us opportunity to become coordinators for the conduct of today's talk. I wish to thank my co-host and coordinator as well, Dr. Monica Arora, who was actively involved in conducting arranging today's webinar. Next, I would like to thank Mrs. Tanushri Chakrabarti, Chief Coordinator, for a kick-start of this program. I would also like to acknowledge Mr. Faisal, who assisted as a technical support on IT infrastructure. Last but not the least, my sincere gratitude to our listeners who have taken out the precious time and attended today's webinar. Hope it was fruitful and benefited you. With this, I, Kalpana Devi, Faculty, Pharmacy, Department of Pharmaceutical Chemistry, would like to sign off and conclude my vote of thanks. Thank you once again. Hope you had a very good talk. Thank you, Tanushri. Over to you, Tanushri.
Thank you, Mrs. Kalpana. So we have come to the end of another exciting lecture of this webinar series. Participants, kindly fill your feedback forms before leaving the meeting. You will be receiving your e-certificates within 5 working days. Our next lecture on this webinar series will be on 30th July. Please note the date for our next webinar series is on 30th July, that is Thursday. The notification and the invitation for the same will be rolled out soon. Till then, be safe, be healthy, stay at home. Thank you all. This is Tanushri from the Department of Pharmaceutics signing out from Al-Amin College of Pharmacy. Thank you all. See you again in the next lecture of this webinar series. Thank you all. Bye. Stay safe.
लीव किया मैंने, बंद कर दो तो चलेगा ना? Not in meeting in progress. I want to leave